Okay, thank you. It is well known that one of the main features for Alzheimer's disease is the loss of episodic memory. Everybody talks that it's a loss of memory related to Alzheimer's disease. This loss of memory could be related to a deficient adult neurogenesis that takes place at the dental gyrus in the adult. This adult neurogenesis takes place from the first proliferation of some stem cells that start to proliferate and to differentiate into new neurons, and these new neurons could connect with the neural network, and the function of this new neural network is related with memory and learning. Also, you know that in Alzheimer's disease, there are two histopathological features: the appearance of the beta amyloid that could aggregate into senile plaques, and the presence of tangles that are composed of tau protein. Then it has been from the beginning of the studies of Alzheimer's disease to try to look for possible connections between these histopathology features and the loss of memory. I mean, if amyloid or tau could be related to the loss of memory. It's well known that there is several connections between amyloid and tau, but the connections between tau amyloid and memory is less known. One connection between amyloid and tau is through one enzyme that is known like GSK3β, mainly because it's the form that is mainly in the brain. We know that amyloid activates this enzyme, and once this enzyme is activated, could phosphorylate tau. In fact, this enzyme is also known like tau kinase 1, and this tau that is phosphorylated is toxic. Then the thing is, this toxicity could be related to some changes in neurogenesis and to changes in memory. Then the objective of our work is to know the consequence of overexpressing GSK3 before and after neuron differentiation during adult neurogenesis. And to do that, we use several models, mice models, conditional models, that we can overexpress the enzyme or downexpress the enzyme using two promoters that express the enzyme. One promoter is expressed before neural differentiation, the other one is expressed after neural differentiation. One promoter is expressed in this type of cells, stem cells, and the other one is expressed probably from here. To test if it really is expressed from here to the differentiation, we tested by immunofluorescence studies the presence of a specific marker for the different types of cells and to see when the reaction against the overexpressed GSK3 appears. And we found that the overexpression from this point and so on continues in the adult neuron. We try to connect this with the loss of memory, as I told you before. And then the first type of experiments that we did is to test if it's a memory impairment due to the overexpression of GSK3 in these conditions. To do that, we used two behavioral methods for the mice that was theoretically with memory impairment. One is the Morris-Water maze that, as all you know, deals with the mouse should remember where is a platform when it's through to this swimming pool, and after several days, the mouse knows perfectly where is the presence of the platform. But in our case, the mouse that is normal, wild type, is doing better than the mouse that is overexpressing GSK3. Then, in this case, it's some kind of memory impairment. Also, but using other tests for memory, the object recognition test, we found also that there is a memory impairment in this type of mouse. Then we have a mouse without memory. And when you lose a function, usually you look if you also lost a part of the structure in the brain. It's something damaged in the brain of that mouse. And in our case, we were looking to the brain of the transgenic mouse, the one is overexpressed in GSK3, and we found that this part of the brain, the dental gyrus, is degenerated a lot compared with the thing that happens in the control wild-type mouse. Then the damage is here. There is less dental gyrus. The decrease in dental gyrus could be due to two things. One thing could be to the death of neurons, and the other thing could be because in this place there is adult neurogenesis, or the formation of new neurons, could be due to the absence of those new neurons. To see if it's a problem of dead neurons, the death, we found that GSK3 could modify proteins like beta-catenin that is a neuroprotector. If it's phosphorylated, it's degraded, and it's no longer neuroprotection. If the other one is that could be phosphorylated is tau protein. And tau protein, when it's phosphorylated, is toxic. The consequence in both cases could be death of neurons. But we were more interested in to know what happens about the adult neurogenesis. To do that, we try to look for new form neurons. And in this case, we use a virus that could express a green protein, and that is only infecting proliferating cells. When we did that, we can infect the proliferating cells and wait several times to wait until these proliferating cells became new neurons and adult neurons. Then, when we infect the dental gyrus, we can find the appearance of new neurons that are colored green neurons, and then we can compare the appearance of these neurons from the wild type that has this form, Y shape, with the shape of the conditional transgenic mouse that has V shape. Then, to do like this, the Anglo-Saxons, to do a long story short, I will tell you the consequence of this. The V cells in the transgenic mouse are not functional neurons. These functional neurons are dead in a short time, and the consequence after the death is microglia is activated, and after activation of microglia, there is secretion by these cells, microglia, of new compounds that could be toxic for the precursor of neuronal precursors. This is indicated here. That is the activation of microglia, and microglia could secrete some new compounds that could kill the neural precursors. If we have less neural precursors in the niche, we will lose the possibility of new neurogenesis. From the molecular point of view, we are now in the middle of the experiments. We know that GSK3 could phosphorylate another protein, NF-kappa B, that could act in phosphorylating form, like a transcription or co-transcription factor that results in a decrease in the amount of a protein called fractalkine that is involved in this change in microglia. And the result in this change is that there is a protein, eotaxin, that will appear afterwards that is increased as a consequence of that. Then, this is a model in theory for Alzheimer's disease. If there is an increase that it could take place, and apparently take place, in the patients of Alzheimer's disease due to an increase in GSK3, we can start to protect, if it's the cause of the disease, using an inhibitor of GSK3. Like we have a conditional mouse and we can express up or down when we like, we can try to know what happens when we decrease, like we use an inhibitor, this overexpression of GSK3. We have reversion, we have the recovery of the memory of the mouse when the mouse is young, but we were unable to recover the memory when it's very old. And the explanation for that is that if we have a depletion of new neurons, because we have a depletion of neural precursors, as I told you before, then we were unable to do a recovery of the memory. Then it's the thing that happens when we use a promoter, the one that was expressed late during the neurogenesis. What happens when we use the other promoter, the one that is expressed early in the stem cells? In this case, we found first that is expressed there in the stem cells, and the consequences are yet the opposite. In this case, we have a larger dental gyrus, and the mouse, or the mice, saw better memory using different paradigms. Then the conclusion I don't know, this is not going. Oh, okay. The conclusion is what happens in the human being, because we have the two differences. Depends when we overexpress GSK3 at the beginning or at the end of neurogenesis. To know what happens in the humans, we tested using the Golgi, Cajal staining, and we found that it seems similar to the first case, when the GSK3 is overexpressed but under calcium calmodulin kinase promoter, and we have in humans cells with a V form. Then we are discussing, and is the conclusion in this case, if we need to use inhibitors for GSK3 or not, to try to use like a target for the disease. For the moment, we think that probably it's not very good because it has another collateral problems. Usually there is no regeneration in peripheral neuron cells and other kind of things. But before end, if I can, and as suggested by Professor Kachaturian, that is looking for new features, for new ideas, or for new things, I will like to only to focus you on one thing. I told you about eotaxin, that eotaxin is something that was increased in all this story. And eotaxin is an aging factor. And all of you know that aging is a main risk for Alzheimer's disease. And this protein is now very famous in some way, or is a fashion protein, because there are several relevant papers in the so-called the vanity journals, those with the very top impact index, indicating that when you I think that the battery is going down. Okay, it's very low. I think that the next speaker should have a new battery to avoid problems. There is something called parabiosis that is to change the blood from a young to an old mouse or vice versa. And the experiments indicated in these papers indicate that when you put blood from young mouse to an old mouse, there is a dramatic increase in neurogenesis in the adult old mouse. And when you look which component of blood is involved, you can see that there are several, like beta-2 microglobulin, but mainly is the one that we just told you is eotaxin. And yes, also to follow a little the talk of the Professor Rosor, he told us that to prevent the disease, there are a clinical trial in Colombia indicating that try to prevent the appearance of Alzheimer's disease in familiar cases. In familiar cases are those cases in which you have a mutation in a gene and you know that with that mutation, if you live enough, you will have the disease for sure. But not only that, they know that with this mutation in one gene, presenilin 1, and a specific that changes a glutamic acid by an alanine, you know that the appearance of the disease is at 44 years plus minus 2. We know even that. Then when they were studying these cases in Colombia and Antioquia, is the thing that Professor Rosor said, they found another part of the family showing that was another population in which the appearance of the disease was 50 years old plus minus 2. It's 10 years later. Then it's the case that should be a protector factor. And when they did a genetic screening to see what happens with these people that develop the disease later, they found that the main change was in the gene of eotaxins that is going down the function of this gene. Then this could be some kind of example of looking new ways to try to at least to delay, I don't say prevent, but at least to delay it and to look for the appearance later of the disease, and that could be enough probably for the moment. And finally, I think that this is the people that did the work, all these people, Feliciana, Maria Llorens, mainly Maria Llorens did a lot of this, and Jerónimo Jurado in this case. And also part of this work and another work was done in collaboration with many groups of CIBERNED. I have some, probably I missed someone, but we have collaboration with more than 15 groups of CIBERNED until this moment. Thank you, I finished. Thank you.